And good evening to everybody. And I think we should start right now because we can't waste time. And in fact, I'm very grateful to my batchmate, Dr. Ambagala. And within few days, that he was quite willingly help us to, do this much needed webinar. And we are in a crisis situation, Dr. Ambagala. And I think there are many requests that we should have this webinar as soon as possible. And on behalf of the Sri Lanka Veterinary Association, I would like to welcome you all for this webinar. And without wasting much time, that this much important webinar, please make use of this opportunity to refresh your knowledge and upgrade your knowledge, as well as and do the needful. And at the outset, I would like to invite Dr. Nimal Jayavira, former Additional Secretary to the Ministry of Livestock, and to introduce our resource person. And over to you, Dr. Nimal.
Okay, thank you, Dr. Ijaz. Good morning, Dr. Ambagala, and good evening, everyone joining from Sri Lanka. And I think all of us are aware that we have an outbreak of African swine fever in the moment. So I think it is very important to have this seminar or the webinar at this time. And maybe we will be able to get a lot of technical information on the disease control measures, preventive measures, etc. So let me introduce Dr. Ambagala. I think many of us know him, but I should do it. Dr. Aruna Ambagala is the section head at the Mammalian Disease Unit and the WOAH, or the OIE Reference Laboratory, Canada. At the section head, he oversees testing services, technology development, research, training and scientific consultation to the National Animal Health Program for high-consequence viral diseases such as classical and African swine fever, blue tongue, episodic hemorrhagic disease, Schmallenberg viral disease, rabbit hemorrhagic disease, pseudorabies, and other emerging viral diseases affecting farm animals. Dr. Ambagala received his Bachelor of Veterinary Medicine degree from Sri Lanka in 1994 and his PhD from the University of Nebraska, Lincoln, USA. He holds a vacant appointment at the Faculty of Veterinary Medicine, University of Calgary, the Department of Animal Sciences, and the Department of Medical Microbiology and Infectious Diseases, University of Manitoba, Canada. So we welcome you, Dr. Ambagala. It's over to you for this webinar. Thank you.
Thanks, Dr. Ijaz and Nimal for the introduction, and thanks for giving me this opportunity. I hope you all can hear me.
Yes, we can hear you.
Yes.
Yes.
Great. Thank you. It's a great opportunity, and I'm so sorry to hear about the situation in Sri Lanka. This is something that we don't want to happen, and we never want it to happen in Canada. And African swine fever, classical swine fever, we are free of those two diseases. And Canada is, as you probably know, is one of the major exporters of pork and pork products. And most of our products are exported to other countries. Almost 70% of our products are going outside. And a disease like African swine fever or classical swine fever will have a devastating effect on the Canadian economy. So we try to keep these two diseases, including many other foreign animal diseases we call the transboundary diseases, away from Canada. So again, I want to thank the SLVA for the opportunity, and thank you all for joining today. I know it's pretty late on Sunday night to listen to my presentation. So before I start talking about African swine fever, I just wanted to give an outline. So this is going to be my outline of my presentation today. I'll talk about a little bit of NCFAD just because I'm sure most of you don't know what the National Center for Foreign Animal Disease is, few slides, and then I will jump on to the African swine fever. We talk about transmission, epidemiology, clinical science, diagnostics, and briefly touch on prevention, control, and summary. As you can imagine, it is a big topic, and I won't be able to cover everything. And all of you know African swine fever. You heard about this, you learn about this in vet school, but this is just to refresh your knowledge, and also to think about what we can do to control the outbreak and hopefully eradicate the disease from Sri Lanka. And again, this is just an initial discussion. We can have several different discussions, webinars down the road to talk about individual topics related to the controls and prevention and things like that. So I'm coming from Winnipeg, Canada, and our lab is part of the CFIA Reference and Research Laboratory Network across Canada. And as you can see, we are in Manitoba. And it is housed with the co-located with the Public Health Agency of Canada under the One Health concept. So we have the scientists working on human health on one side and animal health on the other side. And we have BSL-3, BSL-4 laboratory with large animal facilities. So we can do large animal experiments. We can inoculate animals with different pathogens and collect samples for diagnostic purposes and assay development. And we have WOAH reference laboratories for African swine fever, classical swine fever, highly pathogenic avian influenza, and foot and mouth disease. And also we house FAO reference centers for foot and mouth disease and zoonotic emerging pathogens. So our role is to provide the scientific expertise and technologies for the prevention, detection, control, and reporting of foreign animal diseases, zoonotic infection, and emerging disease. And so we have laboratory services. So we receive samples across Canada from the veterinarians when they suspect a foreign animal disease, including African swine fever. And then we provide, we have a very quick turnaround time. We will send the results almost within the same day to the submitting veterinarians. And in addition to that, we do a lot of research related to these viruses because we don't have a lot of experience here in Canada with these viruses. We don't know how these viruses behave, what kind of diseases they cause. And also we need those clinical samples for the assay development because we develop a lot of assays. We improve the assays that we have daily basis. And for that, we need the clinical samples. We don't have clinical samples here in Canada, so they are all collected from the animal experiments that we perform. And also, as I mentioned, we have reference labs. So there are a lot of reference laboratory activities. So these include, one of the things that we do is training technicians, scientists from other countries related to these diagnoses. And we provide, as a reference lab, we provide diagnostics support if a country needs any diagnostic support related to a foreign animal disease that we deal with, and we provide the training, any advice, and sometimes we have on-site training programs through some funding from Global Affairs of Canada and OIE or WOAH. So the structure that we have is we have 10 units, and you can see like each unit, like similar to what you have in BRI, we have different units dealing with different pathogens. So the second unit is the mammalian disease unit that we have. And then also we have some supporting units like the genomics unit, reagent development unit. We have an animal care unit separately who helps with animal experiments, and we have a QA unit. This is my team, and I'm sure you will recognize Kalhari in the middle here. Okay, jumping into the African swine fever. The forgotten contagion, we call it, because we all know that when we were struggling with the coronavirus infection, African swine fever was creeping through the world. It's a century-old disease. It has been recognized over 100 years ago in Africa. But over the last decade, it claimed over 1/3 of the world pigs, especially after it started moving into Asia. It's a contagious, deadly viral hemorrhagic fever of domestic and wild pigs. The mortality rate can reach up to 100%. It continues to spread worldwide, threatening global swine industry and endangering the wild pig species. And it is a big problem in certain parts of like Malaysia, Indonesia, where they are losing their endangered wild species to African swine fever. And the virus is almost here in the Americas. It was detected in Haiti and Dominican Republic a few years ago. So that it is a big concern for the North Central and South America. It is not harmless to people or any other animals other than the pigs. The transboundary spread impact on trade and livelihood of people is because of the impact on the trade and the livelihood of people. It is reportable to the OIE. I'm sure you remember your virology lectures. In vet school, this is a DNA virus. It is the only known DNA arbovirus. That's the virus that is DNA virus that is spread by insects. The only member of the asfavirus genus in the family of Asfaviridae. The virion has 2 layers of envelopes. It's a highly complex. It's a multi-layered. It's a big virus. Extracellular virion gain external envelope from the host plasma membrane. The two forms, both the intracellular version and the extracellular version, are both infectious. And the genome is huge, large double-stranded DNA. It's about 170 to 190 kilobases. You can imagine the size difference between a COVID virus and this one. This is a huge virus, similar to like pox viruses. It encodes about 170 proteins. The interesting part is 1/3 of the proteins are not essential for in vitro replication. So 1/3 of the proteins that the virus is encoded by this virus is used for the manipulation of the host deviation. So that is one of the main reasons that we have trouble developing a vaccine for this virus. There are 24 genotypes have been discovered across the world, and most of them are present in Africa, except genotype. So this virus is so big that you can even see through a very good light microscope. So this is a light microscope picture that you can see. This is an RBC, and you can see the virus stuck to the RBC. So it's a large virus, and it replicates in the cytoplasm. Unlike other viruses, this virus encodes its transcriptional machinery in the viral genomes. One of the key features of this virus is it is highly resistant. So that's what we are running into. It is the main reason that the virus is spreading across the world very quickly, because it is mostly by humans. We spread the virus across the world, and you can see this long distance spread of this virus is due to the ability of the virus to survive in the meat and meat products. It's a highly resistant virus. Several months in frozen, fresh, and uncooked meat, and even in the salted, dried meat products like ham, the virus will survive for months. And fresh blood, 18 months, and even putrefied blood, 15 weeks, around 15 weeks. And in an animal feed under simulated shipping conditions, they have shown that the virus can survive for about 30 days. There are some recent evidence that the virus does not survive in a lot of feed material, but this was done in an experimental setting, and they show that the virus survived for 30 days. However, the virus can be activated using disinfectants, lipid solvents based on the iodide, phenolic compounds, and hypochlorite. For example, one of the easiest one that we can get access to is bleach. And I'm sure the Vocone is something that probably you use in Sri Lanka as well, and will kill the virus. Changing the pH will kill the virus, and the lime can be used for that, calcium oxide, to bring up the pH, and that will help to kill the virus. And if you cook anything at 70 degrees for 30 minutes, so boiling will definitely kill the virus. And this is very, very important when we talk about swill feeding. And I know it is better to ban swill feeding, and sometimes it is not possible to do that in practical situations where people do rely on those material to feed their pigs, and then we recommend to boil the swill at 100 degrees for at least 30 minutes to kill the virus. So what are the susceptible species? In the sylvatic cycle, as I mentioned to you, this virus is from Africa. In Africa, this did not cause any problem because this virus does not make any of those native pig species sick. Warthogs and bush pigs and giant forest hogs, they get infected with this virus, but no clinical signs developed. Same case with the soft ticks, and we believe that this is a virus from ticks. Ticks originated virus, and soft ticks get infected. But on the other hand, the Suscrofa, the domestic pigs and the wild pigs, the European wild pigs, and including the pigs in Sri Lanka, this is from the Yala National Park. They are susceptible to this virus, and they will get the disease and die. The route of transmission is mostly oronasal, unless ticks are involved, through uncooked contaminated pork products, swill feeding, blood or bloody exudate from infected animals, because if you look at the blood from these animals, they have huge amount of virus in the blood. 10 to the 7, 10 to the 8 virus particles roughly around per milliliter of blood. So you can imagine the amount of virus in these infected animals and the danger of opening a carcass. So you have to take every precaution not to contaminate the environment when you're dealing with these animals. And of course, I mentioned it is a tick virus, and it can be infected through bite from infected ticks. Soft ticks, and I looked at some of the ticks. Of course, in Africa, it's Ornithodoros morbata, and in Europe, there was a case in Georgia, not Georgia, in Portugal, involvement of Ornithodoros ereticus. But in the experimental infections, a number of other soft ticks belong to the Ornithodoros family has been implicated for maintaining this virus in the wild. And unfortunately, we have one of these ticks which are already shown to be susceptible to African swine fever. This species has been detected in Sri Lanka. So we had to be careful with the ticks, with the ASF outbreaks now in Sri Lanka. Okay, so method of ASF transmission, I just wanted to throw this figure because it's very, very important to understand that there are mechanical vectors that can transmit the disease, and also through the mechanical means, the feed, the vehicles, contaminated meat, stable flies, personal items can transmit the virus mechanically. And then the biological transmission is from an infected domestic pig or infected wild pigs coming in contact with a susceptible pig, or if the ticks are involved, and those are biological transmission that can happen. So there are many ways for this virus to move, and we have to think about all possible ways if we have to control the spread of this disease. In Africa, as I mentioned, this virus was happily cohabitating with the ticks, the warthogs in the burrows, and didn't cause any problem until the Europeans introduced the European pigs into Africa, where they started seeing the disease. I'm not going to go into too much details because it's not very relevant to us here. But what's happening is right now is like, so this is the sylvatic cycle that's in Africa between the warthog, the tick, and sometimes other species like the giant forest hogs. But I think what's happening even in now Africa, there's can be another cycle that's between the tick, infected ticks somehow bite a domestic pig, and then this can transmit to the other pigs. So this is possible if the ticks in Sri Lanka somehow get infected, and then they can maintain this kind of a cycle. And then the other cycle is between pigs, and this is this third and fourth ones are anthropogenic cycles, which are purely the third one is anthropogenic cycle. This is by people who will infected pork and infected animal movements. We will maintain this kind of a cycle. And in Europe now, what's happening is the wild boar habitat cycle. And it is possible also happening in Asia, where the virus enters the wild boars, and then the virus maintains in the wild boar population. including the vegetation is contaminated, and then it continued to circulate. So there is possible of having 4 epidemiological cycles of this virus, which started with the first cycle here in Africa. That was the only one that was existing many years ago. So if you look at the global distribution, so there are four continents are affected by now, including you can see Dominican Republic. This was 2023. Unfortunately, at this time, Sri Lanka. Fortunately, at this time, the Sri Lanka was free of ASF, but I think now we will go on the map. Unfortunately. If you look at the history of the virus, there are two situations the virus left Africa. The first one is 1957. That is to Portugal. Again, this was through, they believe it is through the airport. Again, pork products. This is pretty much the human involvement of spreading the virus to Portugal. And then from there, it spread to the Western Europe, all the way even came to Cuba, Brazil, and Dominican Republic and Haiti. But an aggressive campaign eradicated ASF from Europe in 1995, except Sardinia, a small island here. And I think they recently got rid of genotype like last year. It took so many years. You can imagine how long it takes to eradicate a disease from 1957 to 1995. Many, many years of hard work to get rid of this virus. And then after a few years, then 2007, genotype 2 was introduced into Georgia. This is through a seaport. And then it spread to Russia, Europe by domestic and wild pigs. And in 2018, China, which has half of the pig population in the world, it was the worst thing that could happen. And then from there, it spread to many countries in Southeast Asia and India. So that's a global situation of ASF. Looking at the clinical forms of African swine fever, I think that is very, very important as veterinarians for us to know what forms this disease can come to us. So if you look at here in the chart, hope you can see this. There's 4 different forms: per-acute form, subacute form, and chronic form. Per-acute form is caused by, per-acute and acute form is caused by high virulent African swine fever viruses. Subacute form by moderately virulent, and chronic form by low virulent strains. Per-acute form and acute form, you will see like in the per-acute form, but you will see some clinical signs, but sometimes what you will see is just dead of animals. The animals are fine the previous day, you walk into a barn and everything is dead. The acute form, you will see some clinical signs. You will see the fever, appetite loss, lethargy, gastrointestinal, and you will see some hemorrhages, and it takes about 7 to 10 days for the animals to go down. That's probably what you will see in Sri Lanka right now. There's also a possibility of having an subacute form. As the virus mutate and change, there is going to be some moderately virulent viruses showing up. So those will cause less pronounced clinical signs, and the animals will survive animals will live longer with the infection, and also some will recover. Maybe 20%, 30% animals might recover. So this will be really difficult to understand, because it will be a completely different picture from the acute form that you will see at the beginning of an outbreak. And the chronic form is mostly caused by low virulent strains. Those are not very many out there. It will take for a virus to mutate for a while, but there are vaccine strains that are produced which has very low virulent, and those ones, these are moderately virulent vaccine strains, and if they somehow get into a naive population of pigs, and you will see very mild clinical signs. You will not be able to figure out there's African swine fever in there, but you will see some chronic lesions like necrotic skin lesions, pneumonia, pericarditis, abortions, arthritis, and those kind of clinical signs in those animals. And we see that in China, in Vietnam, and in some places, this kind of clinical picture is appearing. As I mentioned, per acute ASF, death of animals suddenly, like overnight, or the acute ASF, you will see some clinical signs like piloerection, huddling, reddening of tip of the ears, loss of appetite. When you go through these clinical signs, you will realize this is not different from any other disease that you will see in pigs. So it's very difficult to say that animals are having African swine fever. Again, erythemic ears, they're cold to touch. You think they're very hot, but when you touch them, they're very cold. These pictures are actually produced at the lab here at the NCFAD by experimental inoculation of animals. So you will see like big cutaneous hemorrhages. They could be petechial hemorrhages to echomatic hemorrhages to like these large areas of hemorrhages on the skin. Bloody feces, and you will see the animals. Sometimes you will see with the constipation, start with constipation, and you'll start seeing some blood, and it can get really worse. And you will see blood-stained orifices. The nasal, the anus will be. You will start seeing some blood tinge areas. When you do the postmortems, you will see a lot of hemorrhagic lymph nodes. So this indicates that the hemorrhagic fever in these animals. It could be anything. Again, it could be PRRS, it could be CSF, it could be ASF. It's so difficult to differentiate these things. But you will see these are submandibular lymph nodes. You can see they're enlarged and they're hemorrhagic. You have to always think, okay, this could be ASF when you see something like this. So this is a cut open lymph node here and here, and you can see they're enlarged and hemorrhagic. These are mesenteric lymph nodes, and you can see they're enlarged and hemorrhagic. Spleen, not always, but you will see splenomegaly. If you remember your textbooks in vet school, they talk about splenomegaly as one of the features of ASF, but it is not always the case. With our experimental studies, we see that not all the African swine fever viruses cause splenomegaly, but it is a good indication that it might be African swine fever. You will see hemorrhages, so this is the hemorrhages in the diaphragm you can see here. And hemorrhagic gastrohepatic lymph node, this is pretty common that we have seen it in almost every cases of African swine fever postmortems we have done. So usually it is so hard to find the gastrohepatic lymph node in an animal, but it is pretty obvious, and you can see in this animal it is huge and enlarged. You can easily, and these are going to be strongly positive if you send these samples to the lab. Gallbladder edema, sometimes you will see, and also you will see hemorrhages of the gallbladder. This is a urinary bladder of an animal, which you can see the hemorrhages. The kidneys, you will see petechial hemorrhages in the cortex. These are a few different cases of African swine fever that we have done. Different virulent strains, not all of them are highly virulent, some of them are moderately virulent, some strains, and you will see hemorrhages in the pelvis. And pericardial effusion, and this is very very strong, and you will see that this is full of blood here. And you can see the hemorrhages in the heart muscles, and again, these are some experimentally inoculated animals in the lab, at the lab. You see a lot of hemorrhages. So pretty much a lot of hemorrhages everywhere in the lymph nodes, in the organs, and the lungs could be very congested and edematous, and you can see interlobular edemas in some of these animals. And you can see a lot of effusion. You will see bloody effusion. Sometimes you see it in thorax, and this was actually from this from the peritoneal cavity. A lot of bloody effusion in the cavities. And sometimes you get these hematomas, and when you open up, you will see it's full of blood. And in sow barns, what you will expect is the miscarriages, abortions, and stillbirths. So just wanted to refresh your memories about ASF and show you some of the examples. But probably you see this in the field when you go. With cases with PRRS, it's so difficult to tell a difference. Classical swine fever, porcine sarcovirus-associated diseases, you will see a very similar picture. There is no unique clinical signs, the postmortem lesions, that I can say, okay, this is ASF, this is not CSF, or this is not PRRS. But you have to always keep it in mind, it could be ASF. And you need to get the samples to the lab for rule out potential African swine fever. And the bacterial, as you all know, there is septicemia that can cause hemorrhages in these animals. Erysipelas, swine dysentery, and also it could be poisoning. Non-infectious causes sudden death of the animals. Rat poisoning, aflatoxins can kill the animals, so it is not always ASF. But because of that reason, you need to get the samples to the lab. So before we talk about the diagnostics that we can do, I just wanted to throw this picture so that you know what happened to the animal when they get infected with African swine fever. So on the x-axis, you will see the days post-infection, on the y-axis, diagnostic results. So if you look at here, you can see the viremia. This is the virus in the blood. But you will see, like, immediately after infection, within days, within 24 to 48 hours, the animals will develop high viremia. So if you take a blood sample from a clinically sick animal, the laboratory can easily detect and confirm that the animal is having African swine fever. But if you take blood for serum, or if you collect serum from these animals, you will not be able to see antibodies, because antibodies will take about 7 to 10 days to develop. So the best sample that you can take from an infected animal that's showing clinical signs with acute cases of African swine fever is the blood sample that you can take from this animal. If it is a dead animal, you can take the tissues for testing for viruses. But if the infection has passed and the animals survive, very unlikely with the acute cases, but some animals, if they survive, they will seroconvert and antibody test can be done. Most of the cases, antibody tests are done for post-surveillance outbreak to regain the freedom that you have to do antibody testing, ELISAs, to show the world, or the OIE, that you got rid of the disease and there are no cases of chronic ASF or low virulence strains are circulating. Because if you have low virulence strains, you will have animals that will survive the infection and then they will seroconvert and you will see them by ELISA. So just so I wanted to show you that how the kinetics works in the ASF infection, so you know why we select certain samples for diagnostics. Diagnostic specimens from the live animals showing clinical signs, whole blood serum. Dead animals, tonsils, spleen is the best organ for African swine fever detection, but you can take lymph nodes that are hemorrhagic or congested. You can take liver samples, kidney sample, extra samples. And if the carcass is, like, if it is already the carcass, it's putrefied carcass, like, for example, what happens with most of the cases with the wild boars, the wild pigs. By the time you find the animal, dead animal, it's already eaten by the others, and you only have the skeletons left. So you can send the large bone. The bone marrow will have the virus, so we can the laboratory can confirm that the animal died of African swine fever. Of course, I don't think I have to explain to all of you, and all of you are familiar how careful you have to be from the collection of the samples and get the samples to the lab, because you can spread the disease, because this virus is highly stable, as I mentioned before, and there's going to be a lot of virus in those samples, thousands and thousands of copies. So you have to be very careful when you ship the samples, and you have to take every precaution not to spread the disease when you send the samples to the lab. Take proper precautions to package properly and send the sample, and make sure the sample is delivered to the lab on time. If you want to know more about African swine fever laboratory diagnosis, this is a WOAH publication that came out in 2024 of May. If you want this, if you don't have access to this, please let me know. I can provide you this. So this is protocols and guidelines for laboratory diagnostic. At different conditions, we talk about outbreak situations, active surveillance, passive surveillance, and for quarantine, what kind of tests that you should be doing, so that you can prepare yourself for the diagnostics needed to deal with the outbreak. Let's move on to the prevention. How do we prevent more cases of African swine fever? Import control. This is very, very important. I'm sure Sri Lanka was doing everything they could do possible to protect the swine industry from import of live animals, pork and pork products, and feed from infected countries. Border security is important, illegal introduction. And I remember there was an incident where this was stopped a year ago, I think, illegal introduction. But it is difficult. It is very, very difficult. That's why the biosecurity is so important for us. No matter how many things we do to control the import, this biosecurity, there's always possibility of illegal introduction. People are traveling from these countries where the African swine fever virus is. They love to bring their pork products. They like their things to be taken with them. When they travel, they like to feel like home when they're outside. So they bring in. I think it is very, very important for us to have the biosecurity and prevent those pork products that contain African swine fever entering our pig farms, whether it's a backyard farms or our commercial farms. So the biosecurity is very, very important, and swill feeding is one of the easiest way that the virus can get into, because the swill, these kind of the food scraps are collected from the airports and the seaports, and this is exactly how the Europe, both the times they got the virus into Europe from Africa. So it's a very, very easy. So it's better to ban swill feeding if possible. But if it is not possible, because we know there is some of the people who have a couple of pigs in their backyard is because they don't have a big income, they really need to rely on the swill, like leftovers from the hotels. But I think we need to encourage them and make sure it's mandatory that they cook and they explain it to them that they have to cook for 30 minutes, boil the samples, boil the swill before you feed it into the animals, because that is so critical, because otherwise not only ASF, you can bring in any other swine diseases from countries. Because remember, most of the countries in Asia has classical swine fever and PRRS and other diseases, and they could spread. They're not as stable as African swine fever, but it is possible that those things, pathogens can also come in to the country. The best example is the recent outbreak of classical swine fever in Japan. After, I don't know how many years, 20 years or something, that they got classical swine fever in Japan very recently, and they have trouble controlling because the virus is in the wild boars. Control, when you have the outbreak, again, this is not going to be very comprehensive discussion, but I just wanted to remind you what are the main important things that we have to do. Immediate quarantine, movement control. And I'm sure that you guys are very familiar with these kind of approaches. You have dealt with avian influenza outbreak, probably in the past, CSF outbreak, PRRS outbreaks in a daily basis. So you know what to do. So immediate quarantine and movement control is very, very important. Infected and dangerous contact premises has to be quarantined, and movement controls. Ban on exit of live pigs, pig meats, and other potentially contaminated material. Stamping out and safe disposal of carcasses. Humane culling of infected animals, plus usually also in contact animals. Avoid risk of further spread by direct or indirect means, because people tend to throw the carcasses into landfills, rivers, and you're giving the chance of scavengers to spread the disease, because this virus is so stable and it will spread. We have a lot of scavengers, the crows, the other birds, and foxes, and you can name many, and those will spread the disease. Water could be a source. So we have to be very careful handling carcasses. Even when burying carcasses, you have to have a sense of like underground water, how deep it is and how closer to these burial areas that when you put carcasses down, that it can leak into the underground water sources. Earth moving equipments, because you will be using a lot of earth moving equipments during an outbreak, and those could be sources of spreading the virus. They recommended lime treated deep trenches, especially in Sri Lanka, where you have a lot of wildlife, and they can dig into these pits, and if it is not deep enough, they will go and pull all those carcasses and spread the disease, spread the virus. So it has to be like very deep when you dig holes to bury these carcasses, because the virus will survive long time, and then can be spread. Proper disinfection, I have listed few of them: sodium hydroxide, any phenol substitutes, and 2% sodium hydroxide bleach, which is easily available and very effective disinfectant for African swine fever. You have to always clean with soap and water before you apply these disinfectants, because most of these do not work in the presence of organic matter. So you really need to clean first before you apply the disinfectants into the farm. And I'm sure you're all familiar with this kind of the approaches for infectious disease control. Intensive active surveillance, frequent clinical examination of pig herds by veterinary officers and inspection teams, good personal decontamination procedures to avoid cross-contamination, because we could be the people who will be spreading the disease if we do not decontaminate our clothes, our shoes. As I said, when you have an outbreak in a farm, that whole farm is fully contaminated. You have to say each and every corner of that farm is contaminated. When we do animal experiments, just environmental sampling, we can find a lot of virus in the pens. So there's a lot of virus these animals will shed, and then they will be everywhere. So you have to be highly cautious, and you have to take every precaution to decontaminate your shoes, your gowns, and everything, and your equipments before you move out of a farm. Traceability of pig is very, very important for disease control. I know this is not something that we may not be equipped with it, but it is very, very important to know where the pigs are moving. The countries like US and Canada, we know each and every pig's where it is moving. Pretty much every pig is registered, so we know where the pigs are moving. I know it is not easy task, but it helps. If you know at least the pig farms, it's very, very important to trace the pigs so that you can go back to the sources and understand where the virus is moving and control. Destocking period and repopulation. So WOAH recommendation is minimum of 40 days after the farm is declared SA positive. You do all the depopulation, you do the proper cleaning. After that, and if there are no ticks involved, and if you don't think that there are ticks involved, because if there are ticks involved in the environment, if the ticks are present, then probably virus will survive longer than 40 days. But WOAH recommendation is to wait for minimum of 40 days before you introduce any animals to the farm. It could be shorter in hot climates like in Sri Lanka, but usually for cold and temperate climates, the 40 days minimum is absolute. It's reasonable, certainly, that these farms will not be reinfected. If you know that these farms will not be reinfected, then you can decide to repopulate, but you have to start with sentinel pigs at approximately 10% of the normal stocking rate of the farm. So 10% of the capacity, you can introduce sentinel pigs and then watch them. Should come from ASF-free zones, and you should watch them for at least 6 weeks. They have to stay free before you can repopulate the farm fully. Even after repopulation, intense active surveillance should be maintained until international declaration of freedom can be made. So if we are going for international declaration of freedom, WOAH, freedom from WOAH, you need to do active surveillance to claim that you're free of ASF. So minimum 40 days is the recommended time. And then after introducing sentinel pigs at the 10% of the normal stocking rate, you have to wait for 6 weeks to know that the farm is free of ASF and you can introduce the rest of the pigs to the farm. If you just look at the factors crucial to the success of an ASF eradication campaign, public awareness is very, very important. We need to inform the people, the farmers. Should be mainly targeted communities affected by the disease. Social media posters, pamphlets, local meetings are the most appropriate means of getting the messages across. There are a lot of material available. If you go to the FAO sites, OIE sites, you can download the pamphlets. You can translate it to Sinhala or Tamil, depending on the region that you need. The campaign should inform people of the nature of the disease, what to do and what not to do. I think it is very, very important what not to do if they see a suspect case, the benefits of getting rid of ASF. So you have to explain to people, it is not the government who will benefit, it is the pig producers who will benefit if you get rid of ASF from the country. This has to be conveyed clearly to the producers. It is their livelihood is in danger if you don't get rid of African swine fever. No domestic livestock species other than pigs is susceptible and the humans are not susceptible. You need to get that information directly to them. Then they don't feel reluctant to handle these carcasses properly, do the decontamination, because I know people will freak out even when they hear the swine flu, that they are worried that they will get infected. I think that message has to be communicated to the people by the veterinarians that they don't have to worry about it. It's a pig disease. A compensation, I'm not so sure how the Sri Lanka will handle this, but I think fairly compensation, farmers should be fairly compensated with the current market value of the animals and goods without delay. Failure to pay adequate and timely compensation will seriously compromise ASF eradication, and I can give you many, many examples for that. It is difficult if the payments are delayed, then people will say, okay, forget it, I'm going to sell my pigs when they get sick. That will create a difficult situation where it will spread the disease further. I want to touch on vaccination. There's no globally accepted vaccine for African swine fever. The modified live vaccines are the most promising to date. So these are live vaccines, as the name implies. So they're attenuated by deleting certain part of the genome of the virus. They are the most promising because I think over the last 50, 60 years, a lot of researchers are trying to develop a vaccine for African swine fever with very minimum success. The subunit vaccines, inactivated vaccines, they do not work against African swine fever. Modified live vaccines are the most promising today. There are two commercial modified live vaccines are licensed in Vietnam. They are highly effective in preventing clinical disease in fattening pigs against the homologous strains, 100% protection almost. However, there are some concerns. The animals develop viremia. They have been shedding of the vaccine virus. There are concerns about reversion to virulence. The virus revert back to the wild type. Recombination, especially when the vaccine is used in the presence of other different genotypes of the virus circulating, or multiple vaccines are used at the same time. There's a potential of recombination. And also there is some concerns about safety in pregnant sows and boars. So these are being studied. They have not proved yet. So we haven't seen any results from these kind of studies, but I know the studies are ongoing to see how safe these vaccines are especially in pregnant sows. Because ASF infected boars will shed the virus in semen. We don't know what the vaccines will do to the in the boars, whether it will shed as well, because they develop viremia. It's not as strong as what you see with the wild type virus, but you will see some viremia developed, mostly intermittent, but you will see some clinical signs sometimes. So depending on, because you have to understand that in experimental setting, most of the times we handle with animals with no other diseases, but in the field situation, when a vaccine is introduced, you have multiple different things going on. The nutrition is different. There are multiple other infections that are going on. So there could be so many other things can contribute to failure of the vaccines. So in China, especially, they have seen multiple different vaccine strains illegally introduced to the field, creating a lot of problems. So WOAH issued this statement a few months ago to watch for substandard vaccines. So this is something that Sri Lanka should pay attention to, because with the outbreak, I don't know how possible it is, but I have seen it in Vietnam, introduction, and even in Thailand, I heard that there are introduction of illegal vaccines. People will bring in and distribute and sell it illegally in the black market, the vaccines. We have seen it, multiple different vaccines in Vietnam, even during, immediately after it was detected in Vietnam. And some of these vaccines really don't work, and that make the condition even worse. So you have to watch for illegal vaccine introduction potential, and the WOAH has warned the countries to be careful when they're using vaccines. Especially, you need to know what is circulating before you introduce a vaccine. I will show you some of the work that we have done to show that these two vaccines in Vietnam are not protective against the currently circulating recombinant strain in ASF. So you have to be really careful if you decide to go for vaccines. And first, you need to know what is circulating. And the other thing is post-vaccination monitoring is so critical, and you have to watch what you're doing, because Vietnam introduced these two vaccines, but there's not real planning for post-vaccination monitoring. None of these vaccines are DIBA vaccines. So serologically, you cannot differentiate from a vaccinated animal from an infected animal. Molecular DIBA assays are there, so by PCR, but it is not always successful, right? Because animals don't develop viremia all the time, and especially with the vaccines. So only during initial stages where the animals develop viremia and do shedding, you can detect them. After many months, it's difficult for you to detect. So this is the study I mentioned. So this was published recently, and our lab was involved in it. So this is in China, they have detected this highly lethal genotype 1-2 recombinant African swine fever virus in pigs. So this is not the 2007 virus. It is the combination of 2007 virus and 1957 virus, genotype 1. Somehow that virus was made into China, and this is a recombinant virus. So we detected it in Vietnam after China declared. So this virus has parts of both genotypes. So it is two different colors represent each genotype. And recently it was detected in Russia, closer to the China border. So we believe it is coming from China. And Vietnam also, we detected it closer to Vietnam-China border. And when we use the two vaccines that are licensed in Vietnam, and we challenge these animals with the new recombinant strains, it is not protecting those animals. So those newly emerging strain of African swine fever is resistant to the two vaccines based on this study to both the vaccines that are licensed in Vietnam. Okay, so I think I'm almost at the end of my presentation. I'm going to leave a lot of time for the discussion. So I just wanted to summarize my presentation. It is not very comprehensive. I just wanted to initiate the discussion, and I want you to think about African swine fever, refresh your memories from the vet school, and think how we are going to control this disease in Sri Lanka and how to protect our industry. It is it's a contagious lethal disease. I wouldn't say it's highly contagious, but it will spread very quickly. It is 100% mortality in domestic and wild pigs. Compared to FMD, African swine fever is not that contagious. Like classical swine fever and FMD is highly contagious. So that's why I said contagious here. But it is contagious enough to kill the pigs and spread, because there's so much virus in the infected animal samples, in the meat, in the blood, in the excreta from these animals. Humans are not susceptible, and not other farm animals other than pigs. ASF is resistant to inactivation and remain viable for long periods in fomites, infected pig meats, and processed pig products. We have to keep in mind it's not the major cause of the spread is humans, not the animals. Clinical signs and postmortem lesions may be confused with the other diseases. We have to always rule out African swine fever and classical swine fever if you see hemorrhagic lesions and the clinical signs I described to you before. They are common to all viral diseases, most of the bacterial diseases, and even non-infectious causes. So you have to do testing. No globally accepted vaccine available for African swine fever. So we have to depend on all other means to control this disease. No vaccine. Even if you have a vaccine is not always the solution. We need to have everything else in place. Vaccine is another tool to control infectious diseases, not the only tool. We know that. So we need to get everything else in place. We need to have a good veterinary structure. We need to have a good laboratory structure. We need to have a good surveillance. We have to have biosecurity. We have to be very vigilant as veterinarians to report the disease, collect the samples, quarantine the farms to spread the disease. It is one of the most, I would say sorry about the typo here, most difficult TADs to eradicate, because the nature of the virus, the amount of virus in these animals, not having a vaccine also contribute. However, the good, there's a hope, because many countries in Europe, first outbreak of genotype 1, although it took 30 years to eradicate, was able to eradicate without a vaccine. But well-organized campaigns can eradicate, and there are examples recently from Europe. Some countries eradicated ASF, depending on how extensive the outbreak is and what we are going to do to control it, will help to eradicate the disease. Some of the points, these again not, I will provide you some additional reading material at the end for you to get a little bit more understanding. But zoning of the country into infected zones, surveillance zones, and free zones is important, so you know where the animals, free animals are, where the virus is. Extensive public awareness campaigns are very, very important. You need to let people know not to worry about it. It's not going to affect them. They have to help to keep the disease away, to eradicate. Enhanced epidemiological surveillance for African swine fever is important, because you need to know where the virus is, where it is moving, to control any disease. Early detection and stamping out by slaughter and proper disposal of all infected and potentially infected pigs with prompt and fair compensation to owners will help for the eradication. Quarantine procedures to contain the disease, including pig movement controls, sale of potentially infected pig products is important. Safe burial of burning carcasses and other infected material is important. When you bury the carcasses, you have to think, like these can be digged out. So you have to prevent any potential digging of these carcasses by the scavengers, and proper burial is important. Proper cleansing and disinfection of infected premises is important. Keeping infected premises without pigs for a safe period. I talk about the rules for the repopulation, and you need to stick to those time periods and the numbers, so that you know that you are not providing pigs as a source for the virus that remains in the farm. Even if you do complete disinfection. There are some corners in some areas that you can't clean, right? So you have to always think that possibility of having some virus in the farm. So 40 days will help for the virus to die, and especially in Sri Lanka, I think with the weather, the temperature will help us to kill the virus. But it is better to stick to the 40 days if possible. Additional resources, there's terrestrial manual. These are all free online resources that you can read about the disease, African swine fever. And if anybody has problem accessing these documents, please reach out to us. And there's a really good free resources that you can download. This is from FAO. They talk about the biosecurity, culling and disposal, clean chain approach for African swine fever, especially smallholder settings, because in Sri Lanka, I imagine that we have a lot of smallholder farms or backyard farms. So some of these tips will help to control monitoring and surveillance of African swine fever. So there is so many resources available because of the ASF outbreaks in Asia already. I think we have access. They prepared all these documents that will help you to plan the control program. As vets, I think I just wanted to put this one up. This is also coming from FAO and OIE. We can spread the disease spread. As veterinarians, we have a huge role to play here to protect our pigs, our farmers in Sri Lanka. So recognize the clinical signs. You can see the clinical signs again. There's no ASF specific clinical signs that you can pinpoint and say, okay, this is ASF. But look for these signs. If you see these signs, always think that it could be ASF or it could be classical swine fever, PRRS. There are some listed diseases here. So you have to always have that in mind that it could be African swine fever, and make sure you get the samples to VRI as soon as possible so they can do the diagnosis and confirm, so you can do the quarantine. If you suspect ASF, I would say like you do the movement control immediately, because now you have an ongoing outbreak. I'm not so sure the legal system will allow you to take these actions, but in Canada, I think we can. The CFIA has the legal authority to stop movement of animals if you suspect, and to put a farm on quarantine until we get the diagnostics done the next day. And the samples are again very, very important. We need to confirm the diagnostic, because laboratory test is the only way we can say it is African swine fever. Collect it aseptically, wash your hands before and after collection. And I told you that you don't want to be the person who is spreading the disease. Handle it carefully, avoid sample degradation, contamination. You have fridges available, freezers available. You can put the samples in the fridge if you can't get the samples to the lab immediately. Bag, seal, package, label it according to the laboratory guidelines, and make sure you are transporting the material carefully, protected. Dispatch it to the diagnosis lab as soon as possible. And notification, collecting the epidemiological information is important. You have to notify this is CFIA. It is in Canada, it is a law that everybody, including farmers, can go to jail if they don't report any foreign animal disease in Canada. So veterinarians, the farmers, everybody is legally bound to report foreign animal disease, which makes it very easy for reporting for us. We will know immediately. Collect epidemiological information. This is important for the veterinarian to do. Quarantine, alert the neighboring farms, and avoid visiting other farms at least for 48 hours, because your clothes, your boots, and everything could be contaminated. So at least wait for 48 hours, and that probably will help. And you need to take a full body shower and things like that. But still 48 hours minimum to stop spreading the disease. That's all I have for you. And I have my email address at the end. And thank you again so much for this last minute, very quick webinar. And I think we have enough time if you have questions. But if you don't have questions immediately, you can always reach out to us. And we are happy to help if you have any questions. So thank you so much.
Yeah, thank you so much, Dr. Ambagala, for very elaborative and informative presentation with a lot of technical details and covering almost all the important aspects. So yes, very happy that you are going to be here with us for the time. And yes, this is the question time. So you can use the hand raise option if you want to ask, or you can put any questions in the chat box as usual. So, okay, we'll see. Dr. Palika, you can ask your question.
Thank you very much, Dr. Ambagala, about at this critical situation, helping us in this way. Actually, I want to ask how many months the virus will be in the long bone after that buried.
I don't know whether there are. I think it could almost up to a year. You can find the virus. I'm not so sure exact time, but I'm sure many months the virus can be found in the bones. So
Yeah,
when the full
now,
carcass, yeah.
Now our people are burying in not in a proper way. Probably they are not putting some lime or some disinfectant like that. So nowadays people are burying all the carcasses within their premises. So I'm doubt whether we can get rid of the virus from those premises within a short period, as you mentioned, in 40 days.
I think once you bury them, if the scavengers don't pull them out, I think the virus will degrade. There are some studies done in Europe where with the wild boars, even like wild boar carcasses that are left outside for months. What they are seeing is that they see DNA, but they don't see any live virus. I think if you bury them properly, I'm not so worried about if they bury them deep enough. I think they will go through putrefaction and it will kill the virus within few months. But if this is pulled out, I think it's a different story, right? Immediately after burying and within few days, if it is the carcasses are pulled out, that's what I'm suggesting. Bury them like very deep, so that the animals cannot bury these the carcasses and pull them out. And you have to be careful not to bury these carcasses closer to the wells and the rivers and things like that, because when it starts leaking, and those things can get into the rivers and places like that. So those are the things that I would be careful, and I think so this information has to get to the farmers when they're burying the carcasses. Incineration is better, but I understand it is not possible, and it is not going to be pleasant to burn these carcasses. So burying is probably the best, but as far as you dig deep, and you should be okay.
Yeah, please let me to ask another question. How is about the product after the processed product like sausages and other things? Do you think they are containing viruses? Because we have to stop all the processing if so.
They may contain viruses, but I mean, we can eat it. It's not a problem. As far as the pork products doesn't get to the pigs, it is okay, I think. It is difficult for you to completely stop. I mean, you should avoid sick animals getting into the production line, but it is difficult. I totally understand. Like, I'm sure the other countries, they have these pork products sometimes enter human consumption. Even I'm sure Canada gets a lot of ASF infected meat by travelers and things like that, because our industry is completely separated, very separated from the people. and high biosecurity, that the virus, if the virus doesn't get into the pigs, I think we are safe. So you may not be able to completely eliminate the virus in your pork products, sausages and things like that, but I think you need to inform the farmers and everyone that not to feed any pork products containing things. If swill feeding is done, if you tell them not to swill feed, I'm sure they're going to do it. I think better to tell them like boil 100 degrees for 30 minutes minimum, and then feed that, because that's then in that case, even if you have ASF infected sausages or something in there, it's not going to, virus will be dead. No problem.
Okay.
Thank you very much, Dr. Ambagala. Thank you.
Okay, thank you
You're
very much.
welcome.
And next, we will get the question of Dr. Nimal Chalsiri.
Hello, thank you. I hope you can hear me.
Yes, I can hear you.
Right, okay. Thank you very much for your very simple but a very informative presentation. Thank you very much for that. And I have a question and a suggestion. Before that, I think that Dr. Palika raised out about the burial and how long the virus is in the long bone. I think I think we should educate the farmer. That is the most important thing, whether it is small farmer or large farmer. They should be aware how to get rid of this infected or dead carcass. That is really a farmer education part. But now my question is, I don't know how fair to ask from you this question. What is the actual cause or root of infection in Sri Lanka? Do you know, because you are very interested about this recent outbreak in Sri Lanka? How the vector came into Sri Lanka? Do you have any idea on that? And the other one is, as you pointed out very well, the animal traceability, farm registration, animal traceability, those are very important. Actually, we have been trying to do this for a long time, but when there is a incident like this, the disaster like this, the importance come up. So I think at least now the department should go for the farmer registration, identification, animal identification, and to promote, at least facilitate traceability. So I think this is high time to think about that aspect also. Thank you very much.
Thank you, Dr. Chandrasiri. I have no idea how the virus came into Sri Lanka. This is we are still at the beginning, right? I think it's something that I will have. I had discussions with the VRI, and we are trying to set up some improve the diagnostics, set up diagnostics, and we may not be able to find where the virus came from, how it came from, right? Because most of the times we can't find where these viruses are coming from. Definitely, we know that we didn't purposely bring it in, and so it is somehow got in here. And what I'm hearing is that Sri Lanka has a system of not bringing in any pork products from legally from countries that are affected. That's all you can do. Illegal. I mean, probably you heard there was a huge board of pork meat ended up in US and Chicago. So see, this illegal movement of pork and pork products, it's so difficult to control. Even with all these technologies that we have, we still, I'm sure, even in Canada, we have people bringing in. I mean, with all the notifications, fine, and everything that we have in place to find people who bring in things, I'm sure it is coming in. As far as you have, I think we have to have multi-layered approach, so that we knowing that these will be breached by some people, so we need to have ways to improve biosecurity, prevent the products going in. I totally agree with you. The traceability is so important because all infectious disease control, you need the traceability. Otherwise, you don't know. If you don't know where the animals are moving, where the products are moving, it is very, very difficult to control an outbreak. Yeah, so that is important. I know this is a difficult situation, but like you said, I think let's take this opportunity to make it happen. Bring those ideas up and then have something in place inform the authorities, and we need to have these things to avoid or to control, first of all, to control and eradicate ASF, and then to prevent anything coming in so that we can bring up our animal industry. Because for Sri Lanka it is, and I know it is how important it is to bring up our animal industry, not only the pork industry, the poultry, the cattle industry. So everything depends on this kind of a traceability system. I totally agree with you.
Yeah. The other thing, give me one more minute. Now, Sri Lanka being an island, I think that most of the entry points will be either the airport or seaports. So if we have there are acts and legislations also in Sri Lanka. So if we have pay more attention on those entry points and have a system implemented, at least we could have control this entering this new diseases into Sri Lanka. I think that part has to be strengthened further. Thank you very much.
Yes, I agree. Thank you.
Thank you. Thank you, sir. And next, we will go to Dr. PSC. Dr. PSC.
Hello. Can you hear me?
Yes.
Hello.
Yes.
Dr. Ambagala, I'm Dr. PSC. Actually,
Hello.
In my range that I first start this investigation, because that in 2019 PRRS case reported in our range. That after that in recently this incidents came. Okay, that first came to diagnose, that means came that some disease and some animals dead, and then we sent sample. First it identified as PRRS. Then that clinical science and mortality pattern has changed. Then again we sent some samples. Then only they said that it can be AFS. ASF. Then my question, that both disease can happen at same time, or it is our misdiagnosis or something?
No, both diseases can spread at the same time, right? It's just like us, right? So we can have a cold and we can have something else too at the same time. So it is possible. It is possible to have multiple viral diseases circulating. So that's why it is important to rule out these diseases which have a huge impact, right? These are transboundary disease. So having the diagnostics ready and be able to test, it may look like PRRS, or the next case may look like ASF, but it could be PRRS, right? You don't know. So I think when you have these high mortality, like big incidents, I think it is always good to rule out. Because I've seen some of the postmortem lesions that I received from Sri Lanka during this outbreak. At the beginning, sometimes it is difficult. It is, and I keep telling people, like, by looking at a postmortem, it is difficult to say it is ASF or CSF, and that's why you need the lab. So you need to have the diagnostics, and we need to rule out when you have high mortality and unusual sickness, unusual deaths, to rule out these cases. So it could be CSF. Anything could happen. So
okay, thank you. Thank you
very much. And Dr. Ambagala, let me ask another question which is very related in the chat box. Can be PRRS and ASF, both viral diseases, infected to an animal in same time? Asked by Dr. Chandan. So,
yeah, it is possible.
It's answered now.
Yeah, it is possible.
Yeah, okay. Then we'll go to Dr. Udita Bandar.
Yes, sir. Thank you very much for the presentation. And actually, the same question I wanted to ask about the PRRS and mixed infection. Yes, I have heard it was occurred in India as well. So, what are the things? I think it was like masking effect was there. So, in a mixed infection, Dr. Ambagala, what are the things that we have to consider when we have to control this kind of outbreak?
Well, I think the approaches we take is pretty much the same. First, you have a vaccine, but for ASF, you don't have a vaccine. But everything else you take is pretty much the same approach, right? If you know ASF infected, there's nothing you can do other than stamping out at this point, whether they have a PRRS or whether they have something else. You need to stamp out to control the spread. So you have to take the biggest impact one. First, you may be able to use vaccination to protect the other pigs in the farm, but with ASF that you don't have that option. So you will have to take the most dangerous disease or the disease that you need to control without a vaccine. Will take the upper hand, and then you have to take measures. Everything else, like the biosecurity, movement control, everything is important, even if it is a PRRS outbreak, so it will control. So these are common things that you take with any outbreak. So, but other than that, I mean, if you have ASF in the farm, then you will, unfortunately, you will have to stamp out.
Well, there's
Okay, thank you very much.
a interesting question in the chat box about treating swill by acidifiers. Can you know?
I am not so sure about it. I'm sure you can, but I don't know how palatable it is after you use acidifiers to swill. Boiling itself will probably destroy the taste that the pigs like it. Maybe boiling is much easier. There's a possibility that things like citric acid can inactivate African swine fever. There are some chemicals that can inactivate, but how effective in a situation where you have a swill, everything mixed up, and sometimes, you know, like these things cannot reach the products. Like if it is a sausage, I mean, it will reach the surface, but it will not reach the inside of the sausage, probably where the virus is. When you cook it, I think you will cook it and then temperature will reach 100 degrees. So I would prefer to stick to cooking if you have to do the swill feeding.
Okay, thank you. Dr. Susil?
Right. Thank you very much. Thank you very much, Dr. Ambagala. That was a wonderful presentation. I learned a lot from that. And very simple question. I'm glad that we ban any imports from infected countries, ASF infected countries, even meat and bone meal, which is coming from bovine origin, have been banned in Sri Lanka to import from ASF infected countries. In case, if we are to check at the ports, how sensible it is to check virus at the ports, and also how likely that we miss the virus if we take samples from large containers of say pork to detect the virus. Is it advisable to do that?
Thanks for the question, Cecil. I think, yeah, I wouldn't advise because it is impossible, and you can't get a representative sample. And we started doing some work, and even in Canada, we don't test samples at the airport, and we assume that there are some positive samples coming in. There's no way you can test each and every sample that's coming into Canada through the airport. People bring in, and like how many cases you stop, right? Not every case you can stop, and there's going to be probably some meat is entering the country. I think the best thing we can do is to make sure that the swill is boiled and fed to the animals, and also to inform the biosecurity and try to do those kind of things. It is difficult, even if you find something. What are you going to do, right? It could be a dead virus, because mostly what we are doing is PCR testing, and PCR only measures the DNA. That doesn't mean there's live virus in it. So there may be a lot of DNA that's floating around, and especially testing like a feed container, and how do we know where the virus is hidden? So those are impractical. I think some of the things that North America is doing is to hold the feed for 30 days, because there's so you hold it for 30 days. So if there's any virus, it will be dead by the time it is released from the port. So, so those are the things that you can do to prevent any potential introduction through these means to the country. And of course, I think separating and informing the farmers is very, very critical.
Can I intervene? Actually, I can remember Professor Tatil. He was when he was our statistical teacher. He was mentioning about this, how to take a sample. It should be a representative sample from a 40 feet container. There are ways and means to do that. So therefore, we need to worry about that if we follow those instructions. So that is done by all over the country, and there are very simple. Can't be that simple, but there are ways and means to get a representative sample from a container. So that won't be a big question. Thank you.
No,
Yeah, so,
Dr. Chandrasekhar, if it is for a chemical analysis, yes, but this is completely different. We are dealing with infective material.
Yeah, that's the thing. That's for chemical analysis, yes, you can do a representative sample, but for these kind of things, you don't know where the virus is. It is not going to be evenly distributed in a container, and even what's the practical point? Like, even if you detect the DNA, is it telling us anything? No, because countries like Japan, I don't remember what other countries, but they have done this testing, and they know the virus is coming into the country. We know, I'm sure, even in Canada, we know this is coming. It is difficult to control people. We know how difficult to tell people, and they won't listen to you. So there is virus coming in, but as far as you have other measures, that doesn't mean that you have to let people bring in anything they want. You need to put the restrictions in place. You need to have the fine and everything. And then of course you have to bring in the feed and everything from countries which is free. We have to do all that, but that doesn't guarantee that the virus entering the country. There's no 100% proof that by having all those things in place will completely prevent a virus entering the country. So we have to have the other steps taken to prevent the virus reaching our pigs. So that's what I was trying to say. Like, even in Canada, we don't waste our time testing the samples that are taken at the airport for testing for African swine fever. We don't waste our resources for that because it doesn't tell us much.
Okay, such finding will be important if they are going to be positive only, no?
Even if it is positive, it could be dead virus, and then you have to prove that you have to isolate the virus. Isolating African swine fever is difficult. It is not easy. Even if you find it, like, that's one sample that you tested. What are you going to do with the next passenger?
Yeah. Okay.
It is difficult. I mean, you have to put fines. You have to definitely impose fines. You have to have restrictions. We have detector dogs in the airports. They are trained to detect pork products, and they go through the luggages, and if they find it, they will be fined, and then all those things will be incinerated. But those are the things we can do at the airport.
Okay. Okay. Next question from Dr. Kamalik.
Thank you very much, Dr. Ambagala. I think you have already answered one of my questions. I thought to ask, it is multiple infections can take place at the same time. I have heard that one before also, because I'm from the diagnostic laboratory. We sometimes come across sometimes BVD and IBR becoming positive. Like this case also. Initially, clinical presentation died more towards to PRRS, and then it went up to hemorrhagic condition in organs. A lot of bleeding in the organs, and clinical presentation was changed. So then it became ASF, African swine fever. My another question is regarding carrier pigs. If a pig is infected with ASF, if it becomes carrier pigs, how long virus will persist?
Thanks, Kamalika, for the question. Yeah, so for the first comment you made, yes. So they can have different viruses. You can have multiple. We have received samples from Africa, some places where it is positive for ASF, and also when you do the whole genome sequencing, mostly shotgun sequencing, we will see different viruses in the same animals. So it is possible to have multiple infections at the same time. Regarding the carrier animals, this is a controversial question related to ASF, and we know the animals can once they get infected, you can have intermittent. If they recover, especially from a moderately virulent strain, they can be viremic for some time. One of the things I did not mention is with African swine fever, unlike other viral diseases, there are no neutralizing antibodies. So there are antibodies produced, but they don't neutralize the virus. The virus is so smart that it can evade the neutralizing capability of antibodies. So there's no neutralizing test for this virus. For that reason, so as far as the animals stay viremic, although they have antibodies present, it could be infectious. How long? These are animal experiments. It's very hard to do. You can imagine doing animal experiments in containment. It's very expensive, so we can't keep animals going forever. But we know at least few months the virus can be shed and could be infectious. They have shown with genotype 1 up to 3-4 months, those animals can be infectious when they infect the animals with some of the genotype 1 strains of African swine fever, and then they bring in contact animals, and then they will see that these animals get infected. So it could be months. Exactly how long, we don't know.
Okay, thank you. Other thing is, if a sow is infected, whether the piglets can become persistently infected.
No, not like CSF. They won't. But you can cause abortions, mummifications, and things like that. But the colostrum, if the sow survives, the colostrum will not protect the piglets because, as I said, they don't have neutralizing capability.
Okay,
Thank you.
Thank you very much. So shall we go to the chat box then for a couple?
Dr. Jayavira, can I ask one more question, please?
Okay, go ahead.
Yeah.
Yeah.
Dr. Ambagala, you have mentioned that ELISA antigen test kit. Can we use that ELISA antigen test kit for diagnosis? And what is the specificity and sensitivity of that? And what is the cost? I want to know those things.
Yeah, no, very, very important question. When I said ELISA, I was promoting the antibody ELISA, but there are antigen ELISA kits available. They're not as sensitive as the real-time PCR. So we have tested multiple different lateral flow assays because we are looking at some countries who doesn't have even diagnostic labs. Like we have countries that we are dealing with in Central America, South America, that they don't have the ability. For example, Haiti, looking at the political situation in Haiti, they don't even have a lab but they have ASF. So we were looking at lateral flow assays. But the problem with lateral flow assays is they are so less sensitive. If you don't have the ability to do any testing, I would do the lateral flows. But those lateral flows are mostly antigen-based assays. But if you have the ability to get the samples to a diagnostic lab where they can run PCR, I will stick to that because it's very sensitive and specific compared to antigen ELISAs or lateral flows. They are not as sensitive as, but if you want to increase the throughput, I can find some antigen-based ELISAs. They're not going to be as sensitive as real-time, but they will come pretty close. Because if you have ELISA machines, then you can test it by ELISA. But our main recommendation is real time. But if you cannot manage, you can go to antigen ELISAs where the viral antigens will be recognized. And if you have a lot of ELISA machines, then you can use them.
Yeah. Can we have false positive in RT-PCR?
Very unlikely. Unless you have contamination. Most of the real-time PCR assays are highly specific. I mean, there is a possibility always for anything. So because of that, we have always backup assays. So some things that we will work with VRI as well to establish some of the conventional PCR. We don't use conventional PCR as the first line of screening assay because it takes time. But if we have a positive case, we will go to the conventional PCR just to confirm, especially if you suspect that it is not ASF and you're getting us. If the clinical picture is pretty obvious and your animals are dying and there's epidemiological evidence that is there, then we don't question. But of course, the first case, we will go to conventional PCR and sequencing before we even declare ASF in Canada. But unless you have like a very clear epidemiological evidence, like there's a possibility of dying animals, there's somebody in potential introduction, somebody traveled to ASF infected country and came back, and like all those things. Yes, so to answer your question, even like for real-time PCR, there's a possibility contamination in the lab could give rise to a false positive, right? So you have to always have some sort of a diagnostic assay to back up your real time. But chances are very, very low for real time. It's very specific because you have 2 primers and a probe that are specific to that target. So the chances of you messing up is very little.
Okay, thank you. Thank you, Dr.
Thank you very much. So let me go to the Dr. Disnika's question in the chat box. The countries which have eradicated the disease may have taken firm decisions on depopulation. Countries like Sri Lanka, without proper depopulation actions, vaccination, etc., can overcome this ASF disease. It's a general question. Okay,
Depopulation
you can give a general answer anyway.
Yeah, it is going to be difficult without depopulation because the virus will spread, right? So when you let the virus spread without taking the animals down, I'm not so sure how we can control it. Yeah. It is difficult. You need to have depopulation and movement control. The countries like Vietnam, they started a new method because with time they realized, I think they started with compensation. They realized that it is not feasible, and then they stopped compensation, and then they went to we call tooth pulling. That means you go into a farm, you have infected animals, clinical signs, you only pull those animals out. Yeah. If you do it very effectively, if you have a good veterinary system, very good diagnostic system, you may be able to prevent, because I think what was happening was the farmers were losing animals, and the whole country as a whole, and Vietnam, their main protein source is pork. It's a huge industry in those countries. So they started losing the national herd to ASF. And at that point, the country decided, okay, we are going to do the tooth pulling, where you go and pull the infected animals and trying to save the other animals for protein. So the industry won't collapse. So I don't think it is. At the beginning, they said it's working, but unless you have a good structure where you prevent animals being entered up in the slaughterhouse when they get sick, like many things will play a role to make a mess out of it, right? So just tooth pulling will not do the job. But that is something South Africa is planning to implement. I think they showed that it can work under certain conditions, but it all depends on everything else in the system. So that could be my answer. It's very hard to give a very clear answer for that. Other countries have tried. It might work depending on everything else that is playing around.
Yeah. Okay. Yeah. Dr. Mairuthi's question, in Sri Lanka, one of the important ways for disease spread is through wild animals. In that way, is there any practical method in other countries such as Africa to control the disease through wild pigs?
That is one of the first thing I asked was like, is it in the wild boars? Because getting into wild boars is going to make it difficult for us to control any disease, as we know, like FMD based example. I mean, once it entered the wild pig population, and Europe is having trouble. Germany is struggling with it. The whole Europe, Eastern Europe, is struggling with the wild boar population, with the virus in the wild boar population, and because it tend to sneak into the backyard populations. Right now, there is no clear answer. I know different countries are trying different vaccines, the bait vaccines, to control. CSF was controlled in Europe by bait vaccines in the wild boars. It was possible. ASF, they are trying hard using some of these recombinant MLV vaccines as baits to control. But so far, there has not been very good luck. But you saw in Europe, they used fences. I mean, when the outbreak is contained, they were able to kill the wild boars in that region and control. But at the beginning, they started shooting the wild boars, and then they started spreading the disease, because wild boars are smart. They're very, very smart, and they know this. They know they're very intelligent animals, and when you start shooting them, they will try to get away with it, and then they will spread the disease. That's what happened in Europe. And then later, what they did was they started going from outside to in, and then killing the wild boars. I think that was a little successful, and they could put fences to prevent wild boars, and there was some success in there. But in general, it is going to be tough to control the spread via wild boars. So the minimizing the virus in the wild boar population will help to get rid of this disease.
Okay, to my knowledge, the disease has not been identified yet in wild boar populations.
Okay, that's good. That's good. Then I think it will be easier.
Yeah. Then next question. Is there any possibility of preparing a vaccine against ASF in Sri Lanka? Asked by Dr. Chandran. I think we will have to answer that one. You can give your comments anyway.
Yeah, as I mentioned to you, there's no globally accepted vaccine. The modified live vaccines are out there. There's some licensed vaccines in Vietnam. With African swine fever, one of the critical things one of the difficult things is there's no cell line. There's no cell line that you can use to make vaccines at this point. So most of the countries that they do, except Vietnam, one company which has their own cell line that they have developed. Like one company, they use as primary leukocytes isolated from the pigs, because there's no successful cell culture system for African swine fever. So that makes it even harder to grow these live attenuated vaccine. And we are still learning from all the trials that is being done in Vietnam and in other countries, in Philippines, how safe these vaccines are. So killed vaccines, we know it's not going to work. So if you grow the virus and you use formalin or other things to inactivate, it's not going to work. Subunit vaccines doesn't work so far. I got a pamphlet from one of the meetings one of my colleagues attended in Europe, that China has developed a subunit vaccine, and they claim there's 100% protection, but yet to see. There's a lot of effort going on in other countries, and I think my understanding is China has given up the idea of developing a modified live vaccine because of the issues that they have seen in the field. So they are moving towards a subunit vaccine, multi-antigen vaccine. According to this company pamphlet, it's working, but yet to see the real data. So it's hard to say anything at this point. I would say it is difficult. ASF vaccines is difficult.
Okay, thank you. Thank you, Dr. Ambagala. And Dr. Sanjik is saying that they have identified some deaths in wild boars, but I know that one, but what I mean was the confirmation of the disease.
Not confirmed. Not confirmed.
Yeah,
Yeah.
okay,
Yes.
okay.
Yeah.
And
I hope it's negative.
Hopefully.
Yeah, and Dr. Shant asking, is it advisable to carry out PRS vaccination at this time? Because he's saying that some of the farms are being vaccinated at the moment against PRRS.
I think you should continue your PRRS vaccination, but in the other farms that are not confirmed ASF positive, but again, I think moving to in the presence of African swine fever in the country, you have to take every precaution. You have to understand you will be moving the virus in your vehicles, in your boots, in your clothes, in your equipment. So you have to take every precaution to not to spread the disease. That's why it's so important for you to understand the farms that are affected, confirmed by the VRI, and then so you know that they are ASF infected, they're quarantined, there's no movements of animals, vehicles out of those farms, and then which are the free farms, which are the infected farms. So everything else can continue because you have the pigs to take care of, you have pigs to feed, you have pigs to vaccinate, you have to move the pigs to the slaughterhouses. Everything else can happen.
Okay. Okay, thank you, Dr. Ambagala. All others are appreciation messages. Yes, really, we must appreciate your availability here at this time and the enlightening answers for all the questions. And thank you for the participants as well. And probably in the future, we might need your support very well. And thank you very much for all the support so far by Dr. Ambagala. And so I would like to hand over to Dr. Ijaz for any official of the SLVA.
Yes.
Thanks, Nimal. Ijaz, I just wanted to mention that I have Kalhari. Kalhari, you want to turn on your camera so everybody can see you? So Kalhari works with me, and she's pretty much my right hand here in the lab, and so we are here to help. Anything that we can do with the lab diagnostics, anything, any suggestions, any help. And I mean, me and Kalhari, we work in the lab, but we have a huge team that work on African swine fever. Not that we have it. We are preparing Canada to control it if it shows up here very quickly. We will not use vaccines, so it will be depopulation, detection and depopulation, and get rid of it as much as possible to regain the freedom. I hope it won't happen during my lifetime. Because I don't want an outbreak to deal with, but we are doing everything we can, and we have developed so many tools, knowledge, and we are happy to share to help you to control not only ASF, anything else. We have another team. That's why I showed you, like we have even influencer team, we have FMD team, we have many other teams that anything that we can do as reference lab for WOAH, we are happy to help with the animal industry in Sri Lanka. And so let us know if anything that we can do from here.
Okay.
Yes,
thank you.
Dr. Ambagala, please accept my personal appreciation, and as well as on behalf of the Sri Lanka Veterinary Association. And in fact, there are some requests from the audience that if you could share the presentation, that they can have it again for reference purposes. If you are willing to share, I would appreciate it. And again, thanks a lot within a short period of time that you gave us a very, very comprehensive overview. And then I would like to invite our Assistant Secretary, Dr. Chatura, to wind up the session with the vote of thanks.
Thanks, Ijaz.
Dr. Chatura?
Yep. Thank you, Doctor. And on behalf of SLVA, I would like to pay our sincere gratitude to Dr. Aruna Ambagala. It was a timely needed topic actually, and it was very interesting and very fruitful. So thank you so much, Dr. Ambagala, being part of the resource person today, and also the Dr. Nimal Jayavira being the moderator. And thank you, Dr. Nimal Jayavira as well, and all the participants. And also the special thanks to Dr. Mohammad Ijaz, President SLVA, because that he wanted to get this presentation or the discussion as soon as possible, because that we have, as we all discussed, that we are in a crisis situation in terms of the swine industry. So thank you everyone, and have a good night.
You all.
Thank you.
Thanks, Ambagala.
Yeah, we'll be in touch. Okay,
Yes.
bye.
Bye.
Okay,
Okay, bye.
thank you. Bye.
Thank you.